The structure of DNA. DNA molecules are among the largest known molecules, containing between 1 million and 100 million nucleotide units, so very, very large molecules. And now the nucleotides are joined together in nucleic acids by phosphate groups that connect at the 5' carbon of one nucleotide to the 3' carbon of the next. So you see that if you look here, you have the phosphate or the phosphodiester linkage. It goes from the 5' of one to the 3' of the next one. And you can see that repeats over and over. We have 5' to 3', 5', 3', so on and so forth. And like I said, these linkages are referred to as phosphodiester bonds. And so the result is a chain of alternating phosphate and sugar units to which the bases are attached. Now, the sugar phosphate chain is referred to as the nucleic acid backbone, and that's the part that's highlighted blue in the picture here, and it's constant throughout the entire DNA molecule. So again, this part here, this is the backbone, right? The part here, and you can see that that doesn't change at all. And thus, one DNA molecule differs from another only in the sequence or order of attachment of the bases along the backbone. You see, I have the bases here circled in green. And just like the amino acid sequence of a protein determines the primary structure of the protein, the order of the bases that are found along the backbone provides the primary structure of DNA. The end of the polynucleotide segment of DNA, shown in the picture here, that has no nucleotide attached to the 5' CH2, is called the 5' end of the segment. You see, I have it circled here in green. Now, the other end of the chain is the 3' end, okay? And that's a convention that we write it that way, that the bases along the backbone are read from the 5' end to the 3' end. So we're going to go in this direction. And again, that's a convention. And so the tetranucleotide that we see here is ACGT, not TGCA. Okay, so we always, again, go from the 5' to the 3' and like that. And so the sequence of bases can be abbreviated in this way because the backbone structure along the chain doesn't vary. It's only the bases that change. Okay, example 13.2 says draw the structural formula for the trinucleotide portion of DNA with the sequence AGT. So adenine, guanine, thymine. Abbreviate the structures of the bases just using letters instead of drawing out the whole thing. And point out the 5' and the 3' ends of the molecule. And so let's start by drawing our sugars, our deoxyriboses. So I'll put one here like this, and then it's going to have the phosphodiester linkage. I don't know if I'm going to be able to squeeze all of this on here, but I'll do my best. So then we have our next 5-membered ring like this. We have our next phosphodiester. So again, I'm running out of space here, but let me see if I can move this up here a little bit to make a little bit more space. All right, so now I need my third sugar. So I'll put my third sugar here like that. There we go. So this would be the 3' end where I'm drawing right now. This would be our 3' end here. All right, now I need to finish up these phosphodiester linkages. You can see I didn't draw the entire thing here. And so remember, you need to have a double bond to oxygen and you need to have a negatively charged oxygen on each one. So we have a double bond here, negative charge up here. Then I need to put the missing one here. So we have our oxygen, then we have our phosphorus. And on the 5' end, we're going to have two oxygens with a negative charge. Again, this isn't the prettiest drawing, but you get the idea. So we have our 5' end here. All right, now it said that it goes AGT. Now remember that we go from, right, the way it's written, the convention is we go from 5' to the 3' end like this. And so the way they did it in the textbook, the way they answered this problem, instead of drawing out each one of the nitrogenous bases, the adenine, the guanine, and the thymine, what they do is they just use the abbreviation. So they just use the letter. So I'm going to put them in a different color just to draw your attention to it. So we have A, and then we have our G, and then we have our T, like that. So that way, all you really have to remember is how to draw the phosphate, or sorry, all you have to really remember is how to draw the backbone, and then you can just use the letters as abbreviations. The discovery of the three-dimensional structure of DNA was among the great scientific quests of the first half of the 20th century. And scientists had gathered data that had given clues to some of the properties of DNA, but the ultimate three-dimensional structure remained elusive. For example, scientists knew that DNA contained equal amounts of adenine and thymine, and also equal amounts of guanine and cytosine. In fact, human DNA contains 20% guanine, 20% cytosine, 30% adenine, and 30% thymine. So 20 plus 20 plus 30 plus 30 gives you 100%. Molecular biologist James Watson and biologist Francis Crick were two of the scientists seeking to put many of the pieces of the puzzle together. And Watson and Crick had been working on many different models of DNA structure in collaboration with other scientists, of course. However, critical information from Rosalind Franklin, and you can see her picture right here of Rosalind Franklin. So, critical information from Rosalind Franklin, who was a pioneer in X-ray crystallography, proved to be crucial in unlocking the mystery of DNA's true form. Based on her data and other data, Watson and Crick made the breakthrough revelation that DNA bases, the purines and pyrimidines, were on the inside of a helical structure. And this led to the realization that the actual three-dimensional structure of DNA was, in fact, a double helix. So Watson, Crick, and a colleague named Maurice Wilkins shared the 1962 Nobel Prize for Physiology or Medicine for this work. And Franklin, who again is shown here, deserved to be recognized as well, but no more than three people can share the one award, and they have to be living people. And she had passed away 4 years earlier. Now, getting back to this idea of the double helix has two strands of DNA that are oriented in opposite directions. So, what do you mean by opposite directions? Well, it just means that one DNA strand is going to run from 3' to 5', while the complementary strand is going to run 5' to 3'. And you see that here, right? You look at the top, the purple one goes from 5' to 3', but the blue one goes from 3' to 5', like that. Okay. So those are complementary strands. And we say that the two strands are anti-parallel. Okay. Also, because the base pairs are on the inside of the sugar phosphate backbone, they hydrogen bond with one another. And so all these dashed lines that you see here, these all represent hydrogen bonds. And if you look at this figure shown here, you can see that adenine forms two hydrogen bonds with thymine that I just highlighted in yellow, whereas guanine forms three hydrogen bonds with cytosine, and you can see the dashed lines here, here, and here. So for every adenine on one strand, thymine appears opposite it on the other strand and vice versa. So they always are going to line up. A is always going to line up with T, and C is always going to line up with G. So AT and CG, if you can remember that. And so the base pairs resemble the rungs of a ladder with hydrogen bonds are stabilizing the two strands to the form of the double helix. By understanding how strands of DNA are themselves arranged, the genetic code could finally begin to be unlocked. In fact, the original article published by Watson and Crick in 1953 in the scientific journal Nature states it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. And so the discovery of the DNA double helix was truly among the most significant scientific contributions of the last century. All right, so example 13.3 says one strand of DNA has the base sequence CCATTG. What is the base sequence for the complementary strand? All right, so a base sequence in a DNA strand is always written from the 5' end to the 3' end. So we'll say 5' end. We've got our C, C, A, T, T, G like this, and then we have our 3' end like that. And so the complementary strand, so we'll call this one the original. original strand. And so the complementary is going to go from the 3' end to the 5' end like this. And you remember I said that A always goes with T and C always goes with G. They're complementary. And so we have CC, so we should have GG, and then A goes with a T, and then we have two T's, so we have two A's like this, and then we have a C, and there we go. And this would be complementary, our complementary strand.